Hello, everyone. My name is Shawn Abraham, and thank you for joining me as we learn about bacterial respiration and how we can analyze this phenomena using dynamical systems. What is bacterial respiration? Before we talk about mathematics, let's first talk about the phenomena we're trying to study. Our focus for this lesson will be on bacteria. Bacteria are single-celled organisms that can be good or bad. Some bacteria could be beneficial, like the ones that help break down food. At the same time, some other bacteria are harmful, like the ones that bring diseases. No matter good or bad, bacteria will need to respire in one of two ways. The first way is by aerobatic respiration, where oxygen will be used to break down nutrients. The other form of respiration in a bacteria is anaerobic respiration, where oxygen is not used. In our study today, we're going to be looking into bacteria with aerobatic respiration. In this phenomena, we can see a relationship between nutrients intake and oxygen intake of the bacteria. The system needs to be balanced for the cells to survive, and this is actually something we can model. First, let's talk about what a dynamical system is. Dynamical systems can be used to analyze how phenomena can change over time. With dynamical systems, we can study the outcome of the system with time evolution. This allows us to use differential equations to be able to study and investigate natural phenomena. Using our knowledge of dynamical systems, we can then take a system and study the effects of time to find the rule that the system might follow. Before we are even able to analyze our system, we first need to model our phenomena. Sometimes, this might be very difficult to do in real life events because there could be many different factors to account for. But it is important in our model to consider some states that which our model could take place in. These constant terms will allow us to make the model into a much simpler differential equation. Using a differential equation is vital in math and uses the characteristics of the rate of change as it contains multiple variables as x and y. Dynamical systems can be used to investigate many different types of models in our world. One common model is a pendulum, which uses physical properties of the pendulum and exterior input to create a unique movement that acts like a cycle until it stops. Another example, investigated by dynamical systems, is population growth, which might seem random, but it actually follows a pattern that we can analyze. Even the universe, which is ever expanding, can be modeled using dynamical systems. Using a model, we can form trajectories to see the path the model follows in a space. In order to use dynamical systems to study bacterial respiration, we first need to find a model. Thankfully, we have a model we can use. Here we can see a model we're going to use for our analysis. This model was created by two people named Ferrin and Reladay, and they published it in a paper in 1979. There are two equations we see here to model the balance we talked about before. In our first equation, we have dx dt, which represents the model of nutrient intake in the cell. In dy dt, we see a similar equation, but for the intake of oxygen. We also see some constants in this model. The constants in these equations, A, B, and Q, are constants called sources in the system. These constants allow us to make this model work, and all these constants are going to be positive numbers. Looking at our model, we can see that we'll be using two-dimensional nonlinear equations. Both of the nutrient equation and the oxygen equation rely on each other, and this brings a type of oscillation in the system. As oxygen in the system increases, more nutrients can be broken down. At the same time, the more nutrient intake there is, the more oxygen is needed. Both of these systems cannot be too high or else the system would not work. Our goal for this lesson is to use dynamical systems to investigate the phenomenon of bacterial respiration. We will need to learn how to analyze a two-dimensional, nonlinear system using the model we already have. We can use mathematical methods to break down this task. The first step we are going to do is to find something called fixed points. and use that to identify the classification of the fixed point. Once we've done that, we can continue to use dynamical systems to see how the system would look like in a phase space, using null lines and model the trajectories of our system. Finally, we can study limit cycles and talk about how a model might be a limit cycle. With this case, we can identify conditions of our unknown constants A, B, and Q. Before we start an investigation into this system, let's first define what fixed points are. A fixed point is a state in which a system has no change. As I talked about before, dynamical systems uses different equations which uses a change in time to define a state. If we make this state equal to 0, we can find the points at which there is no change in our system. This is what a fixed point is. We can easily do this by setting our system equal to 0. Here, we can find that x and y values the system needs to produce no change. To begin finding fixed points, let's start by looking at our equations. We know that dx dt is equal to b minus xy over 1 plus qx squared. And we know that dy dt is equal to a minus xy over 1 plus qx squared. Looking at these two equations, we can see a common term between the two equations in the form of xy over 1 plus qx squared. This is a great start, as we can set our equation equal to 0, and we can put both of these equations in terms of this term. What we end up getting is on the right, for b minus x and y, and a. Now that we're able to find a, and that it's equal to b minus x, we can use this to find what x is equal to. Once we get this, we can find out what y is equal to at the fixed point. Plugging this value in allows us to find the fixed points on the right. This is a point that we'll be using to set as our fixed point. Now that we have our fixed point, we can think about how we can analyze this type of point and that we have and how the system acts at this point. It's very difficult to analyze nonlinear systems, especially as we are trying to investigate a system of bacterial respiration. In order to use our fixed point, we need to first linearize our equations. We can do this by using something called the Jacobian matrix. The Jacobian matrix uses a mix of partial differential equations. As we see, it is a set of 2 by 2 matrix with differential partial derivatives in each slot. As we see in the Jacobian matrix formula, we can take a partial derivative of either x or y from both the oxygen and nutrient intake equations. To help out with the lesson, I worked out the math and found the partial derivatives for you to develop a Jacobian matrix. As you can see, we have different equations and the different partial derivatives from the function set. One observation we can make while looking at this matrix is that the equation at position B and D are the same. This might not have a big deal, but it's always important to make observations on any analysis we conduct. Here, I put the fixed point we found earlier and replaced it with all the x and y values at that fixed point. The equation might look like a mess, but we need to remember that a, b, and q are all constants. The next step is to determine what these points could be, and we can use this to find out eigenvalues. Here we have our equation that we'll be using to find out eigenvalues. First, we need to determine the trace of the matrix and the determinant of the matrix. Once we have our values and we have our equation set, we can find our eigenvalues. But in order to even think about finding the trace and determinant, we first need to set values for a, b, and q. For the sake of this lesson, I assumed a, b, and q to be 14 for a, 18 for b, and q being 0.8. Once I did this, I found the trace and determinant and created my quadratic equation. To find the eigenvalues, I found the roots of the equation, and it came out as the first eigenvalue being a positive 9.021155 and a negative 0.15655. Now that we have our eigenvalues, we need to classify it. There's multiple cases that we can use to classify our eigenvalue. In case 1, we can see that both eigenvalues would be real and distinct. If both eigenvalues are positive, we have an unstable node. If both eigenvalues are negative, we have a stable node. If both eigenvalues have a different sign, we have a saddle point. In case 2, we can see that eigenvalues are both real and equal, meaning that there's only really one eigenvalue. If the eigenvalue is positive, we can either have a stable degenerate node or a stable star node. If the eigenvalues are negative, we either have an unstable degenerate node or an unstable star node. In case 3, we can see complex numbers, and that is how eigenvalues are represented. Case 3 uses alpha, which is a real number in the complex equation, as the center. Here, we can see that if alpha is equal to 0, it's a center. If alpha is less than 0, we have a spiral sink. And if alpha is greater than 0, we have a spiral source. Going through our classifications, we can see that our point is a saddle point because both eigenvalues are opposite in signs, and they're both real and distinct. What is a saddle point? Saddle point are semi-stable points. These points are still considered to be unstable, but they're not completely unstable. If we look in the second figure, we can see the pendulum. And we can see that as it goes in the middle, it might be stable. But as soon as it gets pushed in one direction, it's going to turn in that direction, and it won't be stable anymore. In the first figure, we can see that eigenvalues, our eigenvalues specifically, denote two different directions. Here in S1, we can see that S1 is attracted to the fixed point. However, in S2, it's repelling from the fixed point, and this is what the characteristic of a saddle point is. So before we go on, now that we found the fixed points, let's talk about the nullines of the system. We need to use the nullines to find the trapping region of the system. This is a space where all points will be trapped regardless of input point. Nullines can be used to represent the trapping region. To understand what a nullline is, we can start by something very similar to how we did the fixed points. We need to set our main equations to 0. Once we set them to 0, we can find them in terms of y to find out what the equation would be. In the next slide, I've done that for you here. I set x dx dt equal to 0 and found y to be b over x minus 1 plus bqx minus qx. In equation 2, I found y to be a over x minus aqx. I've graphed the equation on the right. And this is how our trapping region will look like using values. Here we have a slope field diagram. Slope field diagrams are used to graph the trajectories of the system. We can use slope fields that we found in order to analyze the system as it travels through space. As we can see, the system will go naturally to this position, no matter what the starting values for x and y are. For example, if we start in an empty space here, it's going to follow the same pattern as we've described. With this pattern, it's going to stabilize at a point in this line. We can see that this is the same for inside the circle and outside the circle. The circle that we're trying to study is this circle at this point right here. Finally, we're going to talk about limit cycles. Limit cycles are an isolated, closed trajectory. In limit cycles, we can see that it is stuck on a pattern that is in the limit cycle points. They are also unique to nonlinear systems, meaning that linear systems won't ever have limit cycles. Our system is a nonlinear limit cycle, and we can think about how the limit cycle might have might have a stable limit cycle. Here on the bottom, we can see three different graphs talking about a stable limit cycle, an unstable limit cycle, and a semi-stable limit cycle. In this stable limit cycle, we can see that all points are being attracted to the main limit cycle. We can see that all points on the inside are being attracted to the line, while all points on the outside are also being attracted to the line, similar to what we have in our phase diagram. In the unstable limit cycle, we have something different. In fact, all points are being repelled from the stable limit cycle line. We can see that all points on the outside go further outside, and all points on the inside go further to the center. Finally, we have a semi-stable limit cycle. Here, we have one half, the outside as in the diagram, going to the limit cycle, while we have the other half, the inside, repelling outside the limit cycle. This could be the same for the opposite direction, as we can have the outside points repelling from the limit cycle, and we can have the inside points being attracted to the limit cycle. Now, for this system, in order to create the limit cycle, we need to make polar coordinates using our system. Due to time, we weren't able to analyze our system this way, but this is what we can use to find our limit cycle. Using polar coordinates with r and theta, we are able to graph our limit cycle to find out at one point will the stable limit cycle be. We can also see the different bifurcation points and see how this limit cycle changes and what factors could change this. Since we have factors of A, B, and Q, we can find out what values of these will create a stable limit cycle. So in conclusion, through this lesson, we talked about how to analyze our system of bacterial respiration while using dynamical systems. Here, we learned about fixed points. In fixed points, we learned how to find fixed points and what they mean. And we understood that there was a change at which there was a point in the system where there was no change. Then we found out how to linearize our nonlinear equation. We needed to do this to find stability at the point. We found out how to do this using a Jacobian matrix, and we found out what a Jacobian matrix was. Once we found the Jacobian matrix, we were able to classify eigenvalues. After we classify the eigenvalues, we could put these into, we can put values of A, B, and Q into this to find a saddle point. We also discussed what a saddle point is and what it means for a system. As we know, these are unstable points, and we understood how this might look like in the space. Next, we talked about phase spaces, and we talked about how to use nullines. We discussed what nullines were and how we can use them to find a slope field diagram. In the slope field diagram, we also found a pattern within the diagram to see how things change. And we can see that no matter what point we start at, it will go back to a certain cycle. This is exactly what we wanted to see. Finally, with limit cycles, we talked about what they were and what the different types of limit cycles are. We talked about what ours might be, but like I said before, we need to analyze this further in order to fully understand what this cycle could be. All in all, we need to we focused on how to study what this limit cycle is. Here I've attached my references to this project that I used to analyze the system. Thank you, and please contact me for any questions you might have.